Hi everyone, this is Scott Reeves, and I'm going to be going through the last of our lectures related to protein, and this one is about amino acid metabolism and protein requirements. And I've gotten information from a few different sources for this lecture. A lot of it is from Stepanek, some of it is from Gropper, some of it is from another textbook that I've used, and then some of the information's from some previous sources that I've used and some new research articles that I've tried to incorporate. So this is a real overview of these topics, and so I'm going to go through relatively quickly, and I'm just kind of pulling out the most important points that I think. And just to kind of reiterate what dietary protein is, well, we need to consume protein because it's a source of essential amino acids. Those are the amino acids that we need, but we don't make in adequate amounts. And then we also need to consume protein because it's a source of nitrogen, and that nitrogen supports amino acid metabolism and the synthesis of other nitrogen containing molecules. The nitrogen is one of the unique aspects of protein compared to other macronutrients. It's also the part of protein that can be harmful. So our body has to use metabolic pathways and excretion processes in order to control the amount of nitrogen in the body, because a lot of that nitrogen can end up as ammonia and ammonia ion, and that's NH3 and NH4, and those can be harmful to tissues, especially the brain and central nervous system. So we'll talk a little bit about that as we go through. Now, one of the reasons that can happen is because it's very common for amino acids to be catabolized. And that means that the amino acid structure is actually broken down. And that involves separating the carbon containing structure, or what's referred to sometimes as a carbon skeleton, from the nitrogen group. So amino acids themselves can be broken down. And that's why there's so much information on this topic, because each of the 20 different amino acids that we use for protein can be catabolized in different ways. So here's an overview of that nitrogen that we were talking about, nitrogen flow through the body. This is kind of an overview of it, and hopefully you can see the cursor when I do this. So we're talking about dietary nitrogen, in other words, dietary protein. That's where almost all the nitrogen comes from, is protein. Enters into the GI tract, goes through the stomach, into the small intestine, where it's digested and then absorbed. And almost all of the dietary protein is digested and absorbed, especially if it's high quality. You're probably looking at anywhere between 85 to 90% of dietary protein is absorbed. And it's absorbed, remember, as either amino acids or dipeptides or tripeptides. Once those molecules enter into enterocytes, they're almost all converted to free amino acids, and those free amino acids basically enter the body and then can be utilized in a variety of ways. For example, proteins can be synthesized using those free amino acids. And this free amino acids represents compartments all through the body, intracellular, extracellular, in other words, in the plasma, inside cellular organelles, all through the body. The largest pools in the body are probably represented by the liver, but also skeletal muscle, because there's so much protein in skeletal muscle throughout the body. And throughout the body, we have protein synthesis occurring, and some of those proteins can actually be secreted or lost in the GI tract, and so some of them are kind of reabsorbed and once again into the free amino acid pool. Some of them are actually lost from the body. Now, we talked about protein turnover. We know that some of those body proteins can be degraded into amino acids, and some of those amino acids can be reincorporated into proteins. And in fact, remember, the major source of amino acids for protein synthesis are actually endogenous amino acids. In other words, those are the ones that are being reused in the body. Now, those free amino acids can be used to generate urea. And urea is the nitrogen-containing molecule that is made in the liver, and it's excreted in the urine. It is the most significant pathway for nitrogen loss in humans and other mammals. So it's very important that our body can synthesize urea in order to minimize nitrogen, the harmful effects of nitrogen in the body. Some free amino acids are catabolized and they generate ammonia, and that ammonia can be incorporated into urea in the liver. And the most significant tissue in the body for this amino acid catabolism is the liver. So that's one of the reasons why I would imagine the urea cycle is there. Now, some ammonia is excreted directly in the urine, but most nitrogen from leaves our body in the form of urea, although we do lose some as ammonia. Okay, now some of those free amino acids are catabolized and they are used to make non-protein compounds such as porphyrins, which are the ring structures in molecules such as hemoglobin. And then when those molecules are degraded, they're converted into things like bilirubin. Other molecules can be degraded into diglucuronide, which both ends up being secreted through the biliary system into the duodenum, through the small intestine, and in the feces. That's how our body's getting rid of some of these molecules. They also are used to make purines, which are nitrogen containing, and creatine. Now the purines are converted to, the nitrogen can be pulled off, and that gives us uric acid, which can be excreted. And then creatine is converted to creatinine in skeletal muscle, and that's also excreted in the urine, so that can be another source of urinary nitrogen loss. Okay, so that's the basics of what we're going to talk about. Now, amino acids in the body, there are 20 amino acids used for protein synthesis. That's what we use for translation. And that's what are recognized by tRNAs that are part of the ribosome translation machinery. But if you were to look at all the chemicals or molecules in the body that have an amino acid structure, there's over 300 of them. They could also be called amino acids. Now, of those 20 amino acids that are used for protein synthesis, 9 of them are essential amino acids. They must be consumed. And the 20 amino acids that are not part of proteins are mostly in that free amino acid pool that was shown on the previous slide. And that's approximately 150 grams in an adult at any given time, with about 400 to 500 grams of flux per day. In other words, about 400 to 500 grams of amino acids are becoming free amino acids and then being used for other molecules as they come out of that pool. Okay, so one of the points of this is amino acid metabolism is very dynamic. It involves every tissue in our body, and a lot of those amino acids were part of proteins that have been degraded. Now they're going to be amino acids that could once again be part of proteins. But this is a dynamic scenario that gives us the ability to degrade and resynthesize significant amounts of protein and other nitrogen-containing molecules. And as part of all of this, so the next bullet point indicates, as amino acids move in and out of the pool throughout tissues and their cells, they pass through membrane transporters. So there's a variety of transporters that, and most work for several different amino acids. So here's a basic diagram of what happens in this case. Some amino acid transporters work by using sodium, and the sodium binds to the amino acid transporter. That increases the carriers of the transporter's affinity for the amino acid. Both the sodium and the amino acid are transported into the cell. The amino acid can be used for protein synthesis or be metabolized, and the sodium is once again transported out by the ATPase, and the process can repeat. Now, there are numerous transporters for amino acids in the body. Most of the transporters move more than one amino acid. And there can sometimes be competition between amino acids for transporters. In general, though, the transporters usually transport the same general type of amino acids, like charged or basic or or hydrophobic, but overall there's a variety of transporters in all the cell membranes, and that's how the amino acids move in and out. And we won't go into any more detail on this, including the regulation or anything. Okay? Now, once amino acids are incorporated in enterocytes, and then they leave the intestinal cell through a transporter, they move to the liver by the portal vein. And the liver is the major site of amino acid metabolism. For example, the liver is crucial for the synthesis of non-essential amino acids, and they are made from other non-essential amino acids. So there's lots of conversions between non-essential amino acids. Most of those types of reactions are by transamination reactions. I'll show you an example in a minute. Okay? Now that can happen in the liver, and then the liver can release the newly synthesized amino acids into the blood, and they can be used by other tissues. Another reaction that's very common among amino acids, especially in the liver, but in other tissues as well, are deamination reactions. Okay? That results in the nitrogen as part of an ammonia group becoming free. Okay? So that's different. Now, when this happens in the liver, the urea cycle incorporates the ammonia into urea, so it can be excreted in the urine. Now, once you pull off that ammonia group, you have a carbon skeleton, or the rest of the amino acids that can be used for things like energy by oxidation. In other words, those amino acids can go into the Krebs cycle. It can be used to generate electrons that can then be used for ETC. So, for example, in scenarios where energy levels are low, a lot of the amino acids undergo oxidation to provide energy for tissues. In situations where glucose is low, a lot of the amino acids become glucose once they lose their nitrogen. In conditions where there's high energy already present in cells, and this is mostly in liver cells, the amine group can be removed from amino acids, and the carbon skeletons from the amino acids can be used to generate fat in the form of fatty acids. Okay, so there's lots of different options for all the amino acids. Now, the liver is also part of the glucose-alanine cycle, which we'll look at in just a minute. Okay, so the liver is the major site of amino acid uptake following dietary intake. So imagine this, you consume a meal that has dietary protein, those amino acids are liberated from the proteins, the amino acids go to the liver. Now what does the liver do with those amino acids? Well, approximately 20% of the amino acids are used for making proteins and other nitrogen-containing compounds. So those amino acids become part of synthetic or anabolic processes. About 23% of the amino acids that reach the liver are released back into the blood without being metabolized, and they enter the general circulation. So they kind of go in and come back out unchanged. However, the biggest fraction of amino acids, approximately 50% of the amino acids reaching the liver, are catabolized. In other words, the amino acid itself is broken apart. The amine group is separated from the carbon skeleton. And each of those components of the molecules have separate fates. So here's an example of transamination reactions. Here's one. This amino acid is being converted to a keto acid product. And so, for example, this amino acid, you can see its structure. When you remove the amine group, it becomes a keto acid. And a classic example in this case would be alanine converting to pyruvate, for example. Now, when you do that, this aminotransferase, which requires PLP, which is vitamin B6, this enzyme takes the amine group and adds it to a second keto acid, and that keto acid becomes a different amino acid. And the classic example here is alanine aminotransferase, which takes alanine, pulls the amine group off so the alanine becomes pyruvate, then adds it to alpha-ketoglutarate, which is the other keto acid here, and alpha-ketoglutarate becomes glutamate, another amino acid. And you can see by this arrow it's bidirectional. In other words, these enzymes can run it either way. So there's lots of different amino acids that can be made and converted through transamination reactions. And by far the most important tissue for this is the liver. So this is the main reason why the non-essential amino acids are non-essential, because they're converted by transamination reactions. A second type of reactions, very common, especially in the liver, are deamination reactions. And this is simply where the amine group is removed and becomes free. For example, here's glutamate. The amine group comes off, and you can see over on the right side of this equation, when you remove this amine group from glutamate, it becomes alpha-ketoglutarate. So it's a ketone. Here's a ketone group. Got a carboxylic acid group. And here is the amine group that at the neutral pH of the cells typically becomes ammonium ion. That's basically the same thing as ammonia. And so if it's in the liver, that ammonium ion can be converted and become part of urea. If it occurs in other parts of the body, there are mechanisms where the tissues try not to accumulate that ammonium ion, try not to release too much of it into the blood as a free molecule, otherwise that is potentially harmful. And so deamination reactions, I would say, are, I guess you could argue not as advantageous as transamination reactions. So here's the urea cycle. This shows the urea cycle where ammonia. So first of all, the urea cycle occurs in the liver. Almost all urea is made in the liver. And the liver can absorb ammonia, take the ammonia, add it to carbon dioxide. That starts the urea cycle, and the urea cycle uses that nitrogen, a second nitrogen that comes in from aspartic acid, another amino acid, and makes urea, which is a water-soluble, non-toxic, nitrogen-containing molecule that actually contains 2 nitrogen atoms that then is excreted by liver cells, goes into the blood, through the kidneys, and is excreted by the kidneys. Okay? Now, the liver can also take amino acids, and remove the amine group and use it for urea cycle, and that would leave a carbon skeleton, and then the liver can use the carbon skeleton for a variety of other things like we talked about before. Can make glucose out of it, can make fatty acids out of it, quite a few different options for that carbon skeleton from amino acids. And most of these amino acids would be coming from skeletal muscle, especially under catabolic phases like starvation or significant stress, where the body is breaking down muscle. A lot of times it's breaking down muscle in order to provide amino acids to the liver so that the liver can support the other tissues. Here's an example of the glucose of a pathway, and this one's called the Glucose-Alanine Cycle. And this involves the liver and skeletal muscle. In this case, what's happening is the liver is able to make glucose and secrete the glucose into the blood, where the muscle tissues take up the glucose, run it through glycolysis for energy. Then when they get pyruvate, they take an amine group from an amino acid, add it to pyruvate. That'll generate a keto acid from the amine group, but it'll convert pyruvate, which is a keto acid, to alanine. So this is a classic transamination reaction in muscle. And now the alanine leaves the muscle and can go to the liver, where the alanine can be deaminated. The amine group is incorporated in urea, and the liver puts the urea into the blood so it can be excreted through the kidneys, and now the alanine carbon skeleton becomes pyruvate, which goes through gluconeogenesis to make glucose, and then the cycle basically continues. Okay, so there's a couple things happening here. First of all, the muscle, when it's in a catabolic phase, or lots of other times when it's just basically breaking down amino acids, rather than putting ammonia into the blood, it's taking the amine group and making it part of alanine. So now the alanine is a safe molecule rather than ammonia, and the alanine goes to the liver and participates in this. And in exchange, the muscle cell is getting glucose from the liver. Okay, so this is a pretty common pathway, always occurring. Sometimes it's more elevated than other times, but this is one of the reasons alanine tends to be one of the amino acids that's higher in the blood, because of this process. Okay, so the liver is a really, really important part or tissue for amino acid metabolism. It's the most important tissue for almost all amino acid metabolism. There tends to be one group of amino acids that are metabolized differently in our body. And these are the branched chain amino acids. And the branched chain amino acids are valine and leucine and isoleucine. And they are called branched chain amino acids because in their side chain, or in their R group, there is a branch. And they tend to be relatively large hydrophobic amino acids. They're used for protein synthesis. They are 3 of the 20 that we use for protein synthesis, but the branched chain amino acids are essential. And there's one of the unique aspects of their metabolism is shown here. Branched chain amino acids tend to pass through the liver and be taken up by muscle. So these amino acids can be used by liver cells for protein synthesis. Sometimes they're used for energy, but they're really not metabolized as highly or as effectively in the liver as they are in skeletal muscle. So here was an experiment they did years ago. A group of investigators fed animals a mixed diet, and the protein source that they used had approximately 20% of its amino acids were branched chains. Okay? And that seems fairly close to what you would expect, because there's 3 of these amino acids out of the 20 that we need that are essential, that are used for protein synthesis. So in this case, they had about 20% of the dietary protein was branched chain amino acids. That's in the portal vein. So they measured that. Then they measured and quantified the amino acids leaving the liver, and they found that approximately 70% of the amino acids leaving the liver were branched chain. In other words, it went from 20% to 70%. And what they realized was that the liver was letting a lot of the branched chain amino acids through without changing them or metabolizing them. So you kind of end up with this enrichment of the branched chain amino acids leaving the liver. And one of the reasons is those branched chain amino acids are important molecules for skeletal muscle. And one of the things that we'll talk about in a second is branched chain amino acids can be a significant energy source for skeletal muscle. And there's also some other features. Okay, remember the 3 branched chain amino acids are essential. We need to consume them in our diet. The liver needs the branched chain amino acids for protein synthesis, but only plays a minor role in catabolizing the branched chain amino acids. Now, under normal circumstances, most branched chain amino acids are taken up and used for protein synthesis and transaminations by the skeletal muscle. In other words, most of their metabolism is in skeletal muscle. Now, transamination can be followed by pathways to yield energy by respiration. In other words, those branched chain amino acids eventually can contribute to Krebs cycle. And electron transport chain. Leucine metabolism also makes signaling molecules, so it has an additional feature to it. Okay, so overall in our body there's quite a few amino acids that are involved in signaling, but the branched chain amino acids tend to be especially important. Branched chain amino acids tend to be used by our body and our cells as molecules that signal protein and important nutrients are intracellular, and that protein synthesis and anabolic pathways can be activated. Okay, so they're kind of molecules that our cells use to kind of indicate or signal that anabolic processes can be stimulated. Of the branched chain amino acids, leucine is by far the most important for signaling. And here are a few things that leucine does. Studies years ago indicated that leucine stimulates insulin release from pancreatic beta cells. So if we consume a diet that has significant amounts of leucine, leucine will go into the beta cells, it will be metabolized to generate ATP, and that stimulates insulin being released. So it's not just glucose that can stimulate beta cell release of insulin. Leucine is also an important effector of that, and we'll talk more about that coming up. Okay, leucine also stimulates muscle protein synthesis. Okay, it does that in a variety of cells, especially muscle cells, and we'll talk about that. Well, actually, I should just mention that leucine tends to stimulate muscle protein synthesis by activating mTOR. Remember, mTOR is the mammalian target of rapamycin. And I said last week that mTOR is a kinase that, among other things, stimulates assembly of the translational complex, especially by helping translation initiation. And that's an important part of this. So one of the things that they've realized is leucine stimulates protein synthesis by an insulin-dependent and an insulin-independent mechanisms. Okay, leucine also decreases muscle protein breakdown, and then we know that it does that, a lot of that, by going through insulin and decreasing activity of the ubiquitin proteasome pathway. Okay. So leucine has unique metabolites that are involved in much of its signaling potential, but overall, I think that it's best to look at it here. Here's a simple slide. Here's skeletal muscle and the branched chain amino acids. So we can see the branched chains, isoleucine, leucine, and valine, and structures aren't shown here. Maybe they should be, but hopefully you can see this okay. Now, one thing to keep in mind is up at the top of these branched chains amino acids can undergo transamination reactions, and that's what these are. So this enzyme right here that converts each of these branched chain amino acids to, in this case, a keto acid. Okay, so this is a branched chain keto acid that comes from isoleucine. This is a branched chain keto acid that comes from leucine. And this is one that comes from valine. Their structures are slightly different. So the keto acid that they make is slightly different. But what's happened is there's an enzyme that is able to metabolize all three branched chain amino acids. And that enzyme is called branched chain amino acid transaminase. Branched chain amino acid transaminase is in significantly higher levels in skeletal muscle compared to the liver. So it tends to be the reason why these branched chain amino acids are metabolized more effectively in skeletal muscle than the liver. Okay? Once the cells have made the keto acid, that keto acid can be metabolized again by this enzyme. It's called a branched chain keto acid dehydrogenase. Okay, and again, same enzyme can work with each of these keto acids. Okay, now you can see that they can go through a series of metabolic reactions, and if we're just looking at isoleucine here, until, in this case, look what they are converted to. Isoleucine can be converted to acetyl-CoA and succinyl-CoA. And these are molecules that are part of the Krebs cycle. So skeletal muscle can take this isoleucine and convert it to Krebs cycle molecules so that they can provide energy. Here's leucine. Leucine goes through its metabolic pathways and can become acetyl-CoA. And here is valine. Valine goes through its metabolic processes and can then eventually become succinyl-CoA to once again provide energy. Now here's an example of how important they are. Leucine is completely catabolized to acetyl-CoA. In other words, all of its carbons become acetyl-CoA. And a molecule of leucine can actually generate more ATP than glucose through its metabolism. Okay? So under aerobic conditions, especially leucine, but the other branched chain amino acids as well, can become good energy sources for our skeletal muscle. Okay? Now there are some other conditions that have to be conducive to that energy production. But overall, that's one of the important factors of these. Now, look at the leucine pathway. Once that transaminase converts it to its keto acid, this keto acid called alpha ketoisocaproate, sometimes it's referred to as alpha ketoisocaproic acid, or we can call it alpha KIC, or just KIC, K-I-C. That molecule, look, there's an alternative pathway. There is an enzyme that is called alpha KIC dioxygenase. That's right here. See the way it's using 2 oxygen atoms, diatomic oxygen. So right here is KIC dioxygenase. It generates HMB. HMB is beta-hydroxy, beta-methyl butyrate. So it's an alternative pathway that's part of leucine. Now, it's not extremely common for this to happen. I think they estimate about 5% of the KIC becomes HMB. The other 95% goes this way. But it does occur. Okay? And this can happen, so that KIC dioxygenase has been found in a variety of tissues. It's in the liver, it's in the kidney, it's in the brain, but it's also in skeletal muscle. Okay? So this pathway tends to be an option in lots of tissues. We're mostly going to look at it in the background or in the context of skeletal muscle. Now here's where it's interesting. HMB is a signaling molecule. So we're going to talk quite a bit about HMB. And in that case, where did it come from? Leucine. And in our context, we're going to say it's leucine that was metabolized in skeletal muscle to form HMB. Okay? Now, here's kind of another interesting, important point. HMB, sorry, I lost my cursor, can also be converted to, here's HMB-CoA, can be converted to HMG-CoA. Remember that? HMG-CoA is an important molecule of cholesterol metabolism. So in this case, what's happening is the leucine could be converted to HMB, and then the HMB to HMG-CoA, and HMG-CoA is sometimes used for cholesterol synthesis. So it kind of gives the cell another way of getting carbons for cholesterol synthesis if that's needed. Okay? Now, leucine signaling. Overall, leucine tends to increase protein synthesis and decrease protein breakdown. At least some of the effects are due to leucine's effect of increasing insulin secretion, right? Because insulin increases protein synthesis, decreases protein breakdown. Leucine also appears to work through mTOR to enhance protein synthesis. So it increases mTOR activation and mTOR's phosphorylation of other proteins. One of the limitations of Thera's research is that specific mechanisms have remained elusive. So they've known for a long time that leucine is a source of energy for skeletal muscle. It's a signaling molecule, and yet they haven't, for example, found out what leucine is directly binding to. However, numerous in vivo and in vitro studies have verified leucine's effectiveness. Okay, so I think we can say overall that a few of the mechanisms for leucine, let me tell you here, let me look through my notes. Okay, so we know that leucine activates the mTOR pathway specifically by activating the P70S6 kinase. Remember, that's the protein that is part of the initiation complex that helps that initiation complex come together, is when that S6 protein of the 43S is phosphorylated. So leucine helps stimulate that part of it. Okay? So leucine tends to increase protein synthesis. Now here's what's interesting about this other part of the pathway. They realized that leucine was directly interacting, but they also realized that leucine is metabolized to a variety of different molecules in skeletal muscle, and they thought that maybe one of those molecules actually was also a signaling molecule. So this is what they found out, that when leucine is metabolized from KIC to hydroxymethylbutyrate, sorry, that's probably, should we say KIC dioxygenase, about 5% of the time we have HMB. And here's a few ways that HMB might work. What they found out in studies, and I don't have these here, but a study that was done, boy, it's been a few years ago now. They supplemented individuals, and these are human studies, and they gave them supplements of HMB, and I think the dosage was something like 3 grams or 6 grams of hydroxymethylbutyrate. Now, one of the questions is, we know that leucine can be absorbed into enterocytes and end up in tissues because we know there's transporters, but apparently HMG can be absorbed through the gut if we ingest it, and it can end up in a variety of tissues, including skeletal muscle. They gave these individuals HMB, and then they put them on a resistance training program. Now, one of the things that happens when you put muscle fibers under strenuous activity or you stress them, their cell membrane will actually break down a little bit and become leaky. And one of the molecules that leaks out is an enzyme called creatine kinase. Okay, that's the one that phosphorylates, helps muscle fibers go back and forth between creatine and creatine phosphate. Okay, so that enzyme is almost only in muscle. So when you start seeing creatine kinase in the blood, you know that your muscle fiber cell membranes have become leaky, they've been damaged, and they need repair. They found that HMB actually decreased the amount of creatine kinase that was released from muscle fibers, and so they hypothesized HMB might be somehow stabilizing the membrane. And they thought maybe one of the ways it's doing it is it's becoming cholesterol, and the cholesterol is being used to kind of reestablish that membrane, and it's helping the muscle fiber actually resist the stress and actually kind of recover from that exercise. Okay. Now, they also were wondering how HMB was it performing some type of signal property, and that's what we've talked about. Now they have kind of realized that HMB works through a few different mechanisms. Well, I'll actually talk about that in a second. Okay. It looks like that HMB functions as a signal. It actually activates mTOR, lost my cursor, mTOR, which then helps protein initiation of translation. And it can have a significant effect. For example, there was a study, this is actually, this is 20 years ago. Can't believe it. I remember years ago when I first started reading these studies, a friend of mine was working at a pharmaceutical company, and she was telling me that they had some clinical data on HMB, and they were using it to actually decrease the effects of cancer cachexia. So they had clinical data indicating that in groups that were undergoing significant stress, HMB actually helped minimize skeletal muscle loss. About that time, they were doing this study where they found they used 3 grams of HMB. It was during intense resistance exercise training. They did it for 3 weeks. They used healthy, untrained young men, and they found that HMB increased lean body mass and it increased strength when they compared it to a placebo-treated group. And there's quite a few studies that have shown that. And I thought it'd be interesting to look at a recent one here. And so I have the reference here for you. This is in the Journal of Physiology, which is an excellent journal. This is 2013, so a few years ago. And this is the effects of leucine and its metabolite HMB on human skeletal muscle protein metabolism. And this was done by 2 main research groups, actually 3, I guess. 2 of them are in the UK, a couple of them are in the UK, and then and there's some people that have been in protein research for a long time, specifically Atherton and this S.M. Phillips here. He's at Phillips is at McMaster University in Canada. So they collaborated on this project to look at the effects of this. And in this study, they used individuals that were, so these are all healthy young men. They're approximately 21 to 22 years of age. They had 15 research subjects, 8 in the leucine group, 7 in the leucine group, 8 in the HMB group. No differences in age or anything between the subjects in each group. Their BMI was approximately 25, so they were recreationally active, but they were not involved in a formal training program, and they participated in this study. They gave them approximately 3 grams of HMB or 3 grams of leucine. Okay? Now, we look down at the bottom here. The subjects were fasting, but exercise was not involved. So essentially all they did was the subjects agreed to be part of the study. They fasted that night. They came to the lab the next morning. They started the experiment, and it was just basically fasting with the addition of either HMB or leucine. Okay? And this is what they kind of found here. So this is what they have. So the branched chain amino acid leucine acts as both a trigger for the initiation of protein synthesis and as a substrate for newly synthesized protein. As a branched chain amino acid, it can be metabolized within skeletal muscle, leaving open the possibility that leucine metabolites might possess anabolic properties. One metabolite in particular, HMB, has shown positive effects on lean body mass and strength following exercise and in disease-related muscle wasting, yet its impact on acute human muscle protein turnover is undefined. Okay? In other words, mechanisms, so far, not really defined. We report here that HMB stimulates muscle protein synthesis to a similar extent as leucine. HMB was also found to decrease muscle protein breakdown. Our observation that HMB enhances muscle protein anabolism may partly or wholly underlie its predefined anabolic, anti-catabolic, supplemental efficacy in humans. So here's, I think, what was the most significant part of this. Now, let me give you some of the mechanisms here. So this is what they found, and let me just, I should load this paper for you. They used a stable isotope system where basically they had these individuals, they put in a stable isotope using phenylalanine that was labeled, and they set up a circulatory system in their leg that allowed them to use this isotope to measure muscle protein synthesis by looking at how much of that phenylalanine was taken out of their solution and that was actually incorporated into proteins during synthesis. Okay? They found that both HMG and leucine increased muscle protein synthesis. Now, it had already been shown that leucine does that, but they were able to show that HMB did that as well in the same manner as leucine. Okay? Now, here's one of the big differences, I think, in this study. They measured changes in plasma or blood, leucine, HMB, and insulin. And they found, for example, when they gave individuals increased amounts of leucine, they found that some of it was converted to HMB. When they gave the individuals HMB, none of it was converted to leucine, which would be as you would expect, right, based on this. Okay? One of the, I think, most interesting findings was that we know that leucine increases insulin secretion, but apparently HMB does not. They gave them HMB and measured plasma insulin. There was no change in insulin. And the reason that's significant is leucine, some of leucine's effects are because of insulin. It doesn't look like any of HMB's effects are because of insulin. I think that's really significant. Okay? So they're finding that HMB enhances muscle protein anabolism, in other words, making protein because of turnover. It's predefined, underlie its predefined anabolic, anti-catabolic supplemental efficacy in humans. Okay? So they found that basically HMG increased muscle protein synthesis. Okay? Now it's also known that HMG decreases muscle protein breakdown by decreasing ubiquitin proteasome pathway. Okay? So, they said, while HMB and leucine both increased anabolic signaling, in other words, both HMG and leucine enhanced mTOR, that pathway seemed to be more pronounced with leucine. For example, there was more P70 S6K signaling activation with leucine than there was HMB. And that's probably because both leucine and insulin stimulate mTOR and P70S6K1 signaling. However, HMB consumption also decreased muscle protein breakdown, and it did it by it changed it by approximately 57%, and it's an insulin independent manner. And I think that was one of the most important parts of this. So one of the questions is, if you supplement, if a person's trying to supplement with this, and I'm not saying just in athletes, but let's say it's somebody that is undergoing cancer, cachexia, or some type of stressful situation where muscle protein breakdown is stimulated, should they consume leucine or HMB or both? And it looks like probably both. Some studies that I also looked at show that HMB might even be more effective than leucine as far as decreasing muscle protein breakdown, and it doesn't rely on insulin, so that can also be very helpful. So I think this is and there's still a lot more work to be done on this. There's still a lot more papers coming out on this, but this is a really interesting part, I think, of amino acid metabolism, how leucine and a specific metabolite is so effective in changing protein metabolism in our body. Okay, so overall, how do we determine protein intake requirements? In other words, how do we know how much protein we should consume? Well, there's a few different ways to do it. There's a factorial method where you're looking at specific aspects of protein metabolism, the catabolism, and how they're used for protein synthesis. But overall, nitrogen balance tends to be the most accepted and practical method, and it's how most organizations that establish recommendations actually determine amounts for their recommendations. In other words, the World Health Organization and the FAO, they both use this. Originally, we took those data and we kind of used them in corroboration with some of ours to determine our DRIs. Since then, more studies have been done in the U.S. and published in the U.S. where basically we use the nitrogen balance for this, for our DRIs. It's done by estimating nitrogen intake. Oops, sorry, missed a parenthesis, which is essentially protein intake versus nitrogen losses. So you measure the amount of protein the person consumes, and then you measure and estimate how much nitrogen they lose, because generally we take it in as protein, as a nitrogen source, but we lose most of it as nitrogen in the urine, in the feces, and other routes of loss, like hair loss, fingernails, sweat, all of that is part of this. Now, in general, if a person's not trying to add tissues like muscle, the amount of nitrogen that they consume should be approximately the same as the amount of nitrogen that they're losing. We say they're in nitrogen balance, and in general, that's where most adults need to be. There's some times in life when we want to be in positive nitrogen balance, which usually when we're growing, during pregnancy, lactation, things like that. But there's not really any times in life where we want to be negative nitrogen balance. Sometimes that happens during disease when we're losing a lot of nitrogen because of catabolic phase. But overall, when we make these recommendations, we say adults want to be in nitrogen equilibrium. And these are findings from a study that was done. This is a meta-analysis that was done, I think, about 2003, a little over 10 years ago, published in, I believe, the American Journal of Clinical Nutrition. And it showed all these data. Each one of these data are results from a study. And this is a relationship between individual nitrogen balances, corrected for dermal and miscellaneous losses, and nitrogen intake. Each point represents an individual's observed response to a specific intake. The median nitrogen requirement is the intake needed for zero balance. Okay, and this was determined to be approximately 105 milligrams per day. 105 milligrams of nitrogen per kilogram body weight per day. So this is 105 milligrams nitrogen. So here's our nitrogen intake. These values right here is 50, here's 100. Okay, this is how much nitrogen they're taking in a day. This is nitrogen balance. So as I just said, if we're in nitrogen equilibrium, we're not trying to gain weight or lose weight or anything like that. We're going to be approximately zero. We want the amount of nitrogen intake to equal approximately nitrogen outtake. So we look straight across here. And if you compile all of these numbers, you end up with the median being at approximately 105. That's what that means. Now, in order to convert nitrogen to protein, remember protein's 16% nitrogen. So if you want to go nitrogen to protein, you take nitrogen multiplied by 6.25. Okay? If you do that, you end up with approximately 0.66 grams of protein per kilogram body weight per day. 0.66. That's the estimated average requirement. Okay? Now, how do you turn that to an RDA? Well, you have EAR. Remember, you either have 2 standard deviations or 2 coefficients of variation in order to cover 97.5% of the population. When you add those 2 standard deviations, here's the RDA. You end up being 0.8 grams per kilogram body weight. There's our RDA. Lots of data indicate that's probably a pretty good protein recommendations for healthy individuals. So here are some assumptions for that RDA. And this is the RDA in the US. I think in Canada it's the same. Some other countries it's not. Here are some assumptions of that. Well, that there's adequate calorie intake. In other words, a person's not eating enough calories. Some of their protein is going to be affected. Some of those amino acids are going to be used for generating energy rather than maintaining protein levels in the body. Okay, so got to have enough calories. It assumes healthy body composition. In other words, kind of a midpoint of healthy body composition. For example, if a person has a lot of adipose tissue doesn't require as much nitrogen to maintain it as skeletal muscle. Okay, so that can affect it. This is sedentary lifestyle. As soon as a person does physical activity that requires their skeletal muscle and other tissues to adapt, and it affects nitrogen metabolism, that can increase protein requirements. And it can be significant. We've talked about this a little bit, but for example, strength athletes might need up to 1.8 grams per kilogram body weight per day. So in some cases, more than double that of sedentary individuals. Okay? The RDA in the US also assumes that it is a mixed diet with adequate amounts of high quality protein. And what do we mean by high quality protein? In very general terms, we mean that protein's amino acid composition has an adequate amount of essential amino acids compared to non-essential amino acids. Okay? Now, in some countries, they don't eat very much high-quality protein, so their protein recommendation is slightly higher. For example, some countries it might be 0.83 rather than our 0.8. So they need a little bit more protein so that the lower quality protein that they're consuming compensates for that. The other kind of variable in high-quality protein is it's got to be highly digestible. And in general, animal products or proteins are more digestible than plants. So we assume mixed diet with an adequate amount of animal proteins that have high digestibility. Okay? So that's how much. Oops. And that is the type of protein that goes into that to determine whether or not a person is maintaining or getting their RDA. So there's another thing to think about here. A lot of times we look at it as protein requirements, but there's also some methods that, other than nitrogen balance, that can be used to estimate requirements for each essential amino acid, not non-essential, but essential amino acids. And they have ways of quantifying the metabolism of each of the essential amino acids in our body. Not all of them are metabolized identically, and they're not all needed, and they are needed in varying amounts for protein synthesis in the body. In other words, some of the essential amino acids are used commonly in protein synthesis. Some of them are relatively rare, like something like tryptophan. It's an essential amino acid, but large hydrophobic side chain, not used by very many proteins because of that, so it's required in lesser amounts in our body and lesser amounts in our diet. And so we can actually look at how many essential amino acids we need. So this is one thing that we should think about. Protein sources should also be considered in their essential amino acid content and profile relative to requirements. This may be used as a means to compare protein sources. If you're trying to figure out how one protein compares to another, which one would be better, let's look at this. And this is from Gropper and Smith, 6th edition. This is in the protein chapter, chapter 6. Here's the essential amino acid requirements. Here's our RDA. So here's tryptophan, histidine. Here's the 9 essential amino acids. Okay. And cysteine and tyrosine are not essential, but phenylalanine is, because we can go from phenylalanine to tyrosine. We can go from methionine to cysteine, so they put that together because these can interconvert. That's why they're like that. But essentially, these are the 9 essential amino acids or molecules that they're made from. Okay. A couple things point out. For example, here's tryptophan. I just mentioned that one, but here are the levels, and these are RDAs. This is milligram per kilogram per day. All right, and here is leucine, the highest. Okay. Now leucine's not stored in high levels. It's not unusually high in proteins, but I think you could probably argue that leucine's probably that high because of its signaling properties, and leucine can also be a significant energy source. But I think it's interesting, look at this. 42 milligrams per kilogram per day of leucine compared to 5 of tryptophan. Now, let's say, for example, I wanted to compare egg whites to quinoa. There's a couple different ways we could look at digestibility. Like I'm pretty sure that egg whites are digested at a higher efficiency than quinoa. The other thing you can look at is you can look at the essential amino acids in each of those and compare them to this. So you can look at the USDA nutrient database, and you could put notes on the essential amino acid amounts of egg whites compared to those of quinoa, and you could compare it to this, just to kind of get an idea of which one of those proteins would be better. And that's why, for example, even though lots of proteins are complete, in other words, a complete protein has all the essential amino acids, some proteins have the essential amino acids in better profiles than others. And I think that's a good argument for what's a good protein source in some cases. So a lot of people don't realize that we can look at it that way, but we actually can. And then, of course, so we can look at proteins that meet all our essential amino acid requirements specifically, and then overall through the day, if we have a mix of proteins that have all of those in an overall good profile, then we can say we're probably consuming a more than adequate protein from good sources. Okay? I think that's it. Here's a summary of amino acid protein requirements. Remember, nitrogen metabolism makes protein unique among the macronutrients. It's the only one with nitrogen, and that allows dietary protein to be a source of nitrogen to make other molecules, but it also means that our body has to metabolize and deal with that nitrogen, unlike the other macronutrients. So the amino acids are required for protein synthesis, but also for the synthesis and maintenance of nitrogen-containing molecules. Amino acids are not present in a storage form. In other words, for carbohydrates, we have glycogen. We could store that in liver and skeletal muscle, or triglycerides we can store as energy. But amino acids aren't really stored anywhere. So if we need amino acids for things like energy or to make glucose, most of them are going to come from skeletal muscle. So we lose a functional tissue or part of it in order to generate amino acids because we don't store them in any other form. Okay, and branched-chain amino acids, especially leucine, exhibit unique roles in metabolism and also in signaling pathways. So I think it is important that we're consuming proteins to consider branched-chain amino acids as in both of those roles, and that'll help us maximize or optimize, I should say, protein metabolism in the body. And I think that's it, so I'm going to stop there. And that'll do it. Thank you.